Hello, welcome everyone to this anticipatory briefing. We are now going to ask ourselves the following question: how to build a human? Our next speaker is the head of the Laboratory of Stem Cells Biology and Developmental Genetics at the Francis Crick Institute. He's beaming in live from the UK. You see him on the screen above me. His studies focus on several different topics: sex determination, development of the nervous system, and the pituitary, which in French translates as hypophyse. And then he also works on the biology of stem cells. He's going to tell us how to build a human. Please help me welcome Robin Lovell-Badge. Hi, Robin.
Hi. Thank you. I'm almost alive from London. I have a horrible cough, which is why I'm.
That's fine. It looks like you're actually in a Swiss chalet or something, a little bit.
It's my home, which is 400 years old, so it's got lots of oak beams and things.
So thank you for being with us, Robin. We hear you well, we see you well, so we're going to try to make it work. I hope you see the room setting, but basically you appear in a 8 meters wide screen above my head, and so you're looking straight into our audience.
Okay,
that is right here. And basically, so, from starting life as a single cell to multiplying into the millions of cells that make up the human body as we know it, new technologies are allowing researchers to shed new light on the fate of cells and on the potential to modify it. And so we're going to specifically dive into a very precise topic. So there's been recently some high-profile scientific papers published, each claiming advances on the previous ones, where structures that closely resemble normal human embryos appear to have been derived from stem cells. Can you tell us a bit your view on this, and how might it be useful for us human beings?
Just to preface my words by saying that, of course, there's a lot of stem cell biology that goes on, which is not controversial. And we know that there are stem cells in many tissues in the body that are there to help that organ, so they're tissue-specific, organ-specific stem cells, to help that organ cope with cells that are lost through normal processes of wear and tear, or to cope with some damage. But then you have other stem cell types, which may be a little artificial in some respects, that are able to give many, many more cell types, different types of cell, so are not confined to single organs. And indeed, when you're talking about pluripotent stem cells, these are cells that can give rise to any cell type in the body. And so there are two types I'm going to refer to. One is embryonic stem cells, which are stem cells derived from very early human embryos. So these are embryos when there are only about 100 cells shortly before implantation. And so these embryonic stem cells have been worked on for a couple of decades now and are clearly very valuable in many respects. The other type of pluripotent stem cell is the induced pluripotent stem cells, which were derived after it was shown, first of all, that you could have human embryonic stem cells. And secondly, it was the demonstration that you could clone from an adult cell back to give something that was representing you could take the nuclei from an adult somatic cell and put those into an enucleated oocyte, and they would give rise to a whole animal. And of course, Dolly the sheep was the famous example of that. So Yamanaka in Japan, working on the premise that means that adult cells have the ability to give rise to a complete animal. He, and using knowledge gained on studying embryonic stem cells and genes required for pluripotency, this ability to give rise to any cell type, he was able to reprogram the adult cells back into embryonic stem cell-like cells, but these are referred to as induced pluripotent stem cells, and they have pretty much identical properties. There have been various labs trying to see what happens if you can coax these early embryonic stem cells to develop in rather than in disorganized fashion, which is what would normally happen if you just put them in a petri dish and let them grow. If you could make them give rise to structures that more closely resemble an embryo. And I'm just going to skip to the last of the important papers so far this year, which was from Jakob Hanna's group in Israel, where on the notion that there are 3 or 4 main cell types that make up the embryo around implantation, which is trophectoderm cells, which are on the outside of the embryo. You have a population of cells called extraembryonic endoderm cells that give rise to the yolk sac, which is another support structure for the embryo proper. And then you have cells called epiblast, which are going to give rise to the embryo, and a few other cell types, such as cells that are going to give rise to extraembryonic mesoderm, amnion and things. And so he basically split his pluripotent stem cells into 3 or 4 different types, 3 groups, and then treated them each differently, slightly differently, and then recombined them in the right numbers. And they were able to aggregate, form little balls of cells. Didn't quite resemble blastocysts, which is the normal human embryo stages, but they had all those relevant cell types present in them. And then he could culture them in systems that allowed these to develop separately, and so individual aggregates, and to expand. And they show this remarkable ability to self-organize, to self-pattern into structures that looked remarkably like early human embryos. And they were able to culture these to a stage which was equivalent to, they said, about a 14-day normal embryo, which is at the time when you have a lot of complexity beginning to develop in the embryo through the process referred to as gastrulation. And so these structures, both morphologically, just how they appear, and molecularly, in terms of the genes that were active or inactive in them, resembled these early human embryos. And in fact, they were pretty indistinguishable from them.
But Robin, can you tell us? So we understand that there are ways now to recombine the stem cells, the different types, together into something that behaves in the same way. Can you tell us a bit, why it might be useful for us?
Obviously the whole idea behind this work is to, or one of the ideas behind this work is to be able to use these as models of normal human embryos. It's very difficult to obtain human embryos to work with in the lab, and of course, in some countries you're not allowed to do this. But even where you are allowed to do it, it's to get numbers that you might need to do a piece of research is very, very challenging. Here, you can, in theory, or even though the methods were not that efficient, you're still talking only less than 5% of the original aggregates go on to give these more advanced embryo-like structures. You could do all sorts of experiments. You could use them because you can start with induced pluripotent stem cells. You can take these from patients who are perhaps having problems with their own embryos developing or not implanting, etc. You can use these to study aspects of why a lot of embryos don't make it doing IVF procedures, in vitro fertilization procedures, why a lot of embryos that begin then fail and you have early miscarriages, to understand congenital disorders, so spinal cord development problems, congenital problems, etc., congenital heart problems, all sorts of issues you could use these embryo models to study, which would be very difficult to use with normal embryos, partly because you just can't get the numbers. Of course, people are very concerned ethically about using normal human embryos that are left over from IVF programs, even if they've been willingly donated by couples for research. So using something which is not an embryo might overcome some of those ethical issues. Jakob Hanna also suggests that you might be able to use these structures as sources of cells eventually for transplantation to deal with the lack of organs for transplants, but that's, I think, a long way off. Now, the issue is that because you're trying to develop, the scientists are trying to develop models of early human embryos, and they want them to be as close as possible to human embryos. So one of the issues becomes, well, at what point would you say, well, they're too close, they are actually human embryos, and you shouldn't be doing the research on them, or you should be governed as if you were doing human embryo research and not stem cell research. And so that's one of the big issues. The other big issue is, well, you've got to know that they're normal. So if they really want to use them as a validated model, you have to validate them against normal human embryos. Now, clearly you can culture these things. The longer you can keep them going, the more likely you are to be able to say, well, that they correspond to normal human embryos or not. But for normal human embryo culture, we have this, in most jurisdictions, we have a 14-day limit for the amount of time you can culture a human embryo, which is just to the point when they begin to undergo this process of gastrulation, which is becoming more complex. So you have a problem at the moment in jurisdictions where embryo research is possible and is regulated. There's either in law or people follow guidelines to say you shouldn't go beyond 14 days. But you would need to, if you're going to really validate the usefulness of these models appropriately, be able to culture normal human embryos beyond 14 days. So that's an issue with respect to governance and regulation of this whole type of research. Should the 14-day rule be extended? That's a big question in the UK at the moment, but also in several other countries beginning to look at this.
So we're going to get questions from the audience. Please feel free to send them using the app. They're going to show up on my screen, but I'm going to continue. When I prepared this session, I learned some new words thanks to you. One is organoids, the other is assembloids. So you'll have to explain a little bit what these are. But so you mentioned that there is rapid progress regarding in vitro derived cerebral organoids and assembloids. And organoids corresponding to different parts of the central nervous system are linked together and potentially to sensory or motor systems such as muscle. Again, can you tell us more about this and how it can be useful? So I guess these are kind of synthetic organs or limbs that you can rebuild.
Yeah. So people are using pluripotent stem cells in particular to derive all sorts of organoids, and these can correspond to many different parts of the body, so the gut, the lungs, et cetera, et cetera. But in terms of what is beginning to concern some people, those derive from parts of the brain, so the cerebral organoids, whether it's the frontal cortex or hindbrain or whichever part of the central nervous system, then people are starting to get a little worried because these structures are getting really quite complex. And so an organoid, cerebral organoid, is simply where you have one part of cells that you coax your stem cells to give essentially one part of the brain. When you have an assembloid, you are linking such organoids derived from different parts of the brain. So it could be from the forebrain and the hindbrain, for example. So you could, or the spinal cord even. So you could have look for connections between those two different types of organoid, and it's become very clear that you get good connections made, so nerves talking to each other in these structures. And then if you hook them up to a motor system such as muscles, so if you've got a bit of cortex, you've got motor neurons, another organoid that's giving rise to motor neurons that will then connect to muscle cells, that you can trigger a response in the first part of the structure, and you can do that electrically or if you modify the cells that you begin with, just shining light. appropriate light onto the structure, and you'll get nerve impulses traveling from the first bit to the motor neurons to the muscle, and the muscles will contract. So people are starting to get worried a little bit about the complexity of these things. And there have been a number of reports on these, which are, scientifically very interesting, because obviously if you can derive these structures, again, from using induced pluripotent stem cells, and you can derive those from anyone. So you can derive them from patients with disorders of the central nervous system. And it's one way of trying to understand how the etiology of diseases, disorders, whether you can use them to discover simple cures for those disorders, whether small molecule cures or genome editing cures or whatever. You could, so potentially very useful for better understanding and potentially to develop therapies. And that is being used. But as I say, you're getting close to something which is behaving like a functional circuit in a person. And then at some point, people are going to start worrying, well, are these structures capable of feeling pain? Are they capable of some form of consciousness? And so you can see that is going to become an ethical challenge at some point. You can also, functionally, they might work better if you take them out of an in vitro system and you transplant them into an animal's brain, because you're going to get real integration also with the various inputs and outputs within the animal brain, and maybe you can measure things even better that way. So then you have even more complications, and people getting nervous about what are you doing to the animal? What about the welfare of the animal, not just of the modified animal itself, but how is that animal going to interact with its parents, with its mother in particular? How's it going to interact with any cage mates? It could be very damaging for welfare perspective if it's behaving differently. And then, of course, if you start noticing things, or people claim that they're seeing things which are more human-like, then that becomes an issue. There has been a paper that got quite a lot of publicity. Montoya, Kagan and colleagues, Friston was the last author, saying, which was entitled, "In vitro neurons learn and exhibit sentience when embodied in a simulated game world." So this is basically showing that you could take human neurons in vitro, connect them up in various ways, such that electrophysiological stimulation and recording cultures are embedded in a simulated game world, mimicking the arcade game Pong. It's a very simple game, but they concluded from this paper that the cultures display the ability to self-organize activity in a goal-directed manner in response to sparse sensory information about the consequences of their actions, which we term synthetic biological intelligence. Now, I think this is worrying, mostly because of the language used in this paper, because there's no evidence that this is really any form of synthetic biological intelligence or consciousness that we really need to worry about. This is simply showing that you can use these structures in the same way as a neural network in a computer. So it's not that, but you have to be very careful with the language in this case, particularly as they referred to these structures or these things growing in culture as dish brains. And I think that highlights some of the issues in this whole field, that you have to be very wary about terminology. So they should never be called brain organoids, because they're not representing a whole brain. They should be called cerebral organoids, or whichever relevant part of the CNS you've taken the original you're trying to mimic. And you should never use words like dish brains. That's awful. And with the embryos models, they're often referred to as synthetic human embryo models. Well, they're not synthetic. They're not synthetic in the way that they've been made out of synthetic materials, or that they've been precisely patterned in any way by human activity, because in that case, they've self-organized. And that's true also of these cerebral organoids. There's a lot of self-organizing and patterning that goes on, which is remarkable. But
so
you see what I'm... Yeah.
So, I mean, so much to unpack. I'm going to try to summarize what I understand, being a total novice to this field. But so, from these stem cells, of which there are different kinds, you've been able, I mean, researchers have been able to kind of recreate human embryos coming with limitations, cannot go beyond 14 days, etc. You also been able to create organoids and assembloids, which would be some kind of systems that can then like rebuild functions or extend functions, be transplanted, even be interfaced with the body of a human or of an animal. And there's a third thing that has been done, which is there's been considerable progress in deriving eggs and sperm in vitro from these pluripotent stem cells, notably in mice. And so it's possible to use these to actually fertilize embryos, all this coming from a stem cell. So can you also, this is kind of the third, like, big thing coming with major questions and applications and ethical issues.
Oh yes, this comes with many ethical issues. So yes, it's been shown by a number of groups, particularly a couple of groups led by Saito and by Hayashi in Japan, but also others, that in the mouse at least, you can begin with either embryonic stem cells or induced pluripotent stem cells, and use these to first of all derive early germ cells, so called primordial germ cells. That's in the mouse, and that's been also done with human pluripotent stem cells, I should point out. You can then coax these further to become early stages of either sperm or eggs, or the cells that are going to give rise to sperm or eggs, by co-culture with somatic cells that correspond to either a testis if you want sperm or ovary if you want eggs. And these somatic cells, they've taken either from animal testes or ovaries, usually in sort of newborn stages. But you can also derive these also from pluripotent stem cells. Now, there are a number of labs, including my own, that have reported the ability to derive gonadal somatic cells, supporting cells, from pluripotent stem cells in mouse and human. So, but anyway, by doing these co-cultures of the somatic cells with the germ cells, it's been possible to get either sperm, or at least spermatids, elongating spermatids, and to get oocytes. And you can fertilize the oocytes with normal sperm, or you can take these sperm-like things and fertilize eggs with those, maybe using intracytoplasmic sperm injection to help it, but you can get fertilization. And these are given live-born animals, live-born mice. There's some evidence that this might be working in non-human primates. It is not yet working in humans, neither to get oocytes nor to get any sperm-like structures, sperm-like cells. But it's going to happen. Everyone tells me it's going to happen. There's lots of issues that need working on, but it will happen. So first of all, why are people doing this? First of all, just as fundamental research into the germline and germ cell development, for which, particularly in humans, we know very little. And it's clear that there are differences certainly between humans and mice in the way that germline develops. And it's probably true between humans and non-human primates, like the macaque as well. Obviously, it would be nice to have a supply of human oocytes in particular for research on aspects of fertilization and early human development. And so that's fairly sort of obvious. And then clinically, one of the big motives is to be able to create sperm or eggs from individuals who've lost their fertility. So this could be where they had a cancer in childhood, and so been treated for that cancer using chemotherapy, radiotherapy, which has rendered them sterile. So because you can make these in vitro gametes, in theory, from induced pluripotent stem cells, you could derive those from the patients, allowing them to have their own genetically related children. So that's a very understandable motivation which are trying to develop this technology. But you start getting, you can get very contentious quite quickly. So you could have genetically related children for postmenopausal women. You could have genetically related children of same-sex parents, in theory. Children from a single donor, that's a pretty horrific thought, but and generally not possible, but one has to think about it. You could have gametes from dead people for whom tissue is available. You can have gametes using cell samples from people that have been obtained without their consent. You can harvest a few somatic cells from people, from blood, or from actually even using a sieve to harvest the cells from urine. You could think of all sorts of opportunities where you might want to do that. Also, just generally, if you are able to derive these in vitro derived gametes, in particular oocytes, you can have many thousands of oocytes, which means that you could do, for example, pre-implantation genetic testing for multiple alleles, whether related to disease risk or to all phenotypic traits. And so that's going to fuel debates about designer babies, which traits, if any, are legitimate targets for selection. You could use these in vitro, or use the stem cells, pluripotent stem cells, for heritable genome editing. In fact, it probably would be the safe way of doing that if you wanted to do that, because you can characterize the edits you've made in vitro before you then turn the cells into sperm or eggs and use them for reproduction. The ability to generate large numbers of human gametes would enable a practice of in vitro human genetics, where you can have multi-generation human genetic studies in a dish, which would be maybe of use to track the impact of various environmental conditions on the development of human disease, or the impact of crossing specific genotypes. So there's some scientific rationale for that. But of course, you could imagine, let your mind wander where that could go to. It'd be pretty horrible. So there's all sorts of issues about safety and efficacy, of course, of how you might be able to show that these in vitro derived gametes are going to be safe enough to use for human reproduction. But we can get into that with questions if you like.
Yeah, I mean, it's incredible. When I listen to you, I can see both the amazingly positive uses of this and the horrific issues that could come out of it. You mentioned, like one-parent babies. I can see some like pathologically narcissist politicians looking into that or stuff like that. It's really both interesting and terrifying, like most major technological advancement. The first question that came in, I'm going to read it. It's from our audience. Evidently, there are many complex factors that go into decisions on the 14-day guidance laws. What approaches would you advocate for? An incremental rolling back of limits, or a rethink on the framework of the overall guidance?
So I think you need to think a little bit about why the 14-day rule was brought in the first place. At least in the UK, it was a compromise between those who wanted to do research on early human embryos and those who objected to it on fundamental principles. And so the 14-day, a limit was suggested because that allowed that compromise. And 14 days was chosen partly because there was a good biological landmark, which was the formation of a structure called the primitive streak. So visually, you could see that was where an embryo got to, but also because twinning can occur anytime up to 14 days. So one argument was that the soul couldn't enter the embryo until after 14 days. Otherwise, identical twins would have the same soul, which is evidently not the case. So that was a compromise used back then. But of course, when that was first drafted or first thought about, it was impossible to culture embryos beyond about 7 days. So it seemed like an impossible barrier to reach. But now we know we can breach that barrier. There's so much research that could be done on embryos between, say, 14 days and 28 days, for where we know very little. It's often referred to as the black box period of development. After 28 days, it's possible to obtain material from terminations. So you can obtain material to work with and get some understanding of that. So a current number that's being thrown about is 28 days, and that's actually also, there's a fairly good morphological marker of a 28-day-old embryo, which is the closure of the neural tube. So the neural tube is formed as a flat sheet, which then folds up, and it zips all the way along the top, and that's complete at 28 days in humans. And there's lots of structures begin to develop during that period between 14 and 28 days, including the neural tube, heart development, et cetera, et cetera. So I think at the moment that would be a very good compromise, but that's my personal view. Some other people are proposing, well, let's make it incremental, let's say 21 days. Others are saying, well, just leave it open-ended, because really it all depends on what can be justified in terms of the importance of the research. If you have a really good research question that required taking up to 35 days, why not? I think that might be okay, as long as you have you should still have robust mechanisms of scientific review, ethical review, and oversight to make sure what's going on, so you know what's going on. I think that would be okay. And that's my general view. What you want is a robust regulatory oversight system that to permit those experiments in jurisdictions where it could be allowed.
So I didn't go too deep into your bio because I would have needed 5 minutes intro because you're active in so many places, but you are obviously active in both public engagement and policy work, notably around stem cells, genetics, human embryo, and animal research. And in ways,
by the way,
science.
involving the public in any of these discussions is so vital. So public engagement on these, and real public engagement, so proper dialogue, is what's important, whether you give members of the public all the information they need to be able to come to some decision themselves, is so important when you're trying to set limits like this.
So, Robin, let's go back to the title of our session, How to Build a Human. So we talked about the fact we could kind of engineer embryos, so today with limitations, but presumably less with time. We talked about building organoids, assembloids, and who knows, maybe down the road, bigger structures like organs. We talked about re-engineering sperm and ovocyte and gamete from stem cells. So where do we stand when it comes to, from scratch, rebuilding a human?
A whole human. Well, as I say, you could begin with your in vitro derived gametes. You can genetically modify those in a way you'd want to. You can then fertilize those to each other, generate embryos, which you can grow to a reasonable amount in vitro, or you could make embryonic stem cells from those embryos and use them to make these embryo-like models. You can do all of this. Growing an embryo from 14 days, beyond 14 days, is, I think, possible, but at the moment, probably not you can't take it very far, because fairly rapidly you come to a problem of, you need a system for efficient exchange of nutrients and elimination of waste products. And we also get oxygen in as well. And of course, we have a placenta when we develop, which does all of that for us. And so how do you establish a placenta in vitro? Well, there are people thinking about this. I'm absolutely not an expert at all on this, so I can't really talk about it. At the moment, the prospect for complete ectogenesis, so developing an embryo into a fetus, into a newborn, entirely outside the body, is impossible. But there are people pushing at both ends. So you might have heard of sheep fetuses being essentially put into this sort of plastic bag-like structures hooked up to appropriate exchange systems for oxygen and nutrients. And that's to help with premature birth. And so those may be going to be applied to, say, help with premature birth, not in sheep, but in humans. So that's pushing backwards from that end. And then these various embryo models and ways of trying to culture intact human embryos longer and longer will probably depend, at least initially, on fabricating appropriate substrates which keep the embryos happy in three-dimensional support systems and with better ways of having sort of an artificial placenta. Now that changes, the placenta changes dramatically during development. So that's going to require not just one system, but multiple systems to be able to do that. But there are people working on this. So.
We have a question which is actually super interesting. Are there any advantages to developing a synthetic organism from sperm and eggs rather than via a Dolly the Ship style nuclear DNA transfer?
Well, yes and yes. Well, first of all, the cloning procedure is not very efficient. So it's still, I mean, cattle, it's more efficient than some other species, but you're still talking about, a few percent only make it to term, and a normal post-term. For these embryo models, again, the efficiency is low, but you can make tens of thousands of these in one experiment. So that's the advantage of these, that it's much simpler technically to make many of them. So I think they have a lot of advantages in terms of studying things in culture, because you can start with induced pluripotent stem cells. You know that they're derived from a particular individual. They will be genetically identical to each other, or pretty much identical to each other. So they're like clones in a dish. The limitation at the moment, which absolutely everyone agrees with, is that these should not ever be transplanted into a human womb. So you should not be allowed to do that. We have no idea what would happen if you did. Of course, people are going to be doing these experiments with animal models. But even if it worked with, for example, a non-human primate, and so far it has not, then if it did, then it's still no guarantee it's going to work with human. So if you can keep that absolute ban there, then I think, well, it's not a ban, it's the prevailing guideline principles that everyone is sticking to, that then I think we don't have to worry about these being used to make cloned people, for example. But we have to be careful of that. And so this is where there needs to be proper thought about how you regulate this area and whether there should be strict laws, how far-reaching those laws should be, how do you deal with jurisdictions that don't make laws in this whole area at all. So it's, there are issues out there.
There are issues for sure, and yeah, it needs to be careful. I can see some positive cases still, like if you could, have Roger Federer DNA and the Rafael Nadal DNA come together into one player, then someone would have a chance to beat Novak Djokovic in the next 20 years. That would be that would be something, the best of both worlds. Thank you so much, Robin, for coming to us from London, straight
I'm not there in
through
person.
Zoom. And we thank you very much for sharing with us all these ideas about what's said. Thanks, that was Robin Lovell-Badge. Round of applause for you. Thanks, Robin.
Thank you very much.
Have a good afternoon. Bye.
Thank you.